lecture scheduled for 1:10, and I'm attending those lectures, so I need to go down to that as well with Dr. Mai. Just the first thing for the MedTrack 1 people and about the clickers, Sean made that announcement in class last hour. I don't know if it was very intelligible, but it is necessary that if you have your clickers, that you get to him those registration numbers. And what I would suggest is, if you've got your clickers, to physically give him your registration numbers in class this afternoon at 1:10, so that you'll be prepared to use those clickers come Monday, if it all works out properly. Yes?
But don't you register online? I think that's what we did, register online and it went through. Isn't that enough, or you still have to give it to him?
Still have to give it to him. Because all of those registration numbers and names need to be inputted into a use list that is part of the program that exists for Turning Point. So your response, if your name and serial numbers or whatever number is not registered on that participant list, it won't be recorded when you try and use that system again next Monday. The other thing, very quickly, one of you asked about the focus of the student seminars. Our two speakers today actually are going to serve as examples for the seminars that should be coming up after them. But kind of in general, depending on the nature of the paper you select, and as instructed by Dr. Tu, you should have him or an appropriate faculty member look at what you plan on presenting, number one, to see if it's appropriate. But his sort of thinking on this is for you guys to select a paper that has to do with some aspect of basic science, and you know, he likes current stuff in the sense that you guys will be taking neuroanatomy, biochem still, physiology, micro, that maybe you select a paper in one of those areas and that would be your presentation, but go to the appropriate faculty member to have that paper looked at to see if it's an appropriate for a presentation like this. If it's a straightforward research paper, what's necessary for you guys to do is to, number one, do your homework in the area. You're going to become the resident expert for whatever area that you're going to talk about, and essentially educate all of your fellow students and the faculty in that area. So I gave a little presentation on biofilms last semester. I was supposed to be the resident expert in biofilms, and so present the nature of it, give some background information, and if it's a straightforward research paper, benchtop research paper where they're doing something in the lab, explain the hypothesis, the question they're trying to answer, and then how they go about answering that question, and then going through the results to see if they did or didn't answer that question. And you should be able to evaluate whether or not it was a good paper after you've done your background work in your reading of the literature in that particular area. So don't select something that you know nothing about, because it's going to be a tremendous amount of work to try and capture all that background information, of course. And the way they're going to be graded is, we'll ask faculty members that have sat in on the presentation to grade your presentation to see how complete it has been. We're going to have faculty members ask you questions, hopefully questions you can answer, just to give us an idea of whether or not you've caught the gist of the paper, that you've gotten that idea through to your audience, the students and the faculty, and whether or not you've put the whole presentation together in a comprehensive fashion, in an understandable fashion as well. So with that, I'm going to introduce our first speaker, who is going to be Maria Bernal. She's going to present the area of wound healing and describe some of the pathology involved in wound healing. This is not a straightforward research paper, so it's going to be kind of a different focus. It's going to be more of an educational sort of presentation of aspects of medicine that you guys have, most of you guys haven't gone through yet.
Thank you, Dr. Hara. Okay, so this is a pathology review, as Dr. Hara said, and my topic is repair, regeneration, and fibrosis. So right here we see that healing is the outcome of injury, and we have an overview of everything that we're going to cover. So there's an injury, there's going to be repair. Within that process, we're going to have inflammation, and repair can lead into complete regeneration of the tissue or fibrosis and scarring. So what is repair? Repair is the balance between matrix deposition and matrix degradation. The matrix is an extracellular substance in which tissue cells are embedded. Right here we see this is asynthetic tissue graft, and we see the cells being embedded into it, acting as the matrix. Okay. There are three key cellular mechanisms necessary for repair. These are cellular migration, actions on extracellular matrix, which include organization, reorganization, and remodeling, and cell proliferation. Cellular migration depends basically on cytokines. Here we have one of the initial steps. It's cytokine migrations, or we can also call them leukocyte migration, and we can also call these extravasation. So here we see the actual monocyte going in between the endothelial capillaries and the basement membrane and exiting into the matrix. Then we're going to have the process of angiogenesis, and this is when capillary endothelial cells are going to migrate through the matrix to form new capillaries. Here we have some key players on this, the vascular endothelial growth factor and the fibroblast growth factor as well, being secreted by the macrophage. So we see how the endothelial cells merge into the actual matrix, forming new vessels. Then we're going to see some pericyte migrations into the connective tissue framework, or a stroma. So just to remind yourselves, pericytes are like mural cells. They're undifferentiated, and they may differentiate into fibroblasts. They support the blood vessels. So here in the picture you see the actual pericyte going into the matrix, or a stroma. Then another step of the cellular migration is when we have this fibroblast migrating into the actual wound site. So everybody is going right there to the injury site. And the last step of cellular migration is going to be re-epithelialization, and this is when epithelial keratinocytes detach from the neighboring cells and basement membranes and migrate between the scab and the wound, as seen in the picture. The second mechanism of repair is going to be the actions of the extracellular matrix. So it is, and the extracellular matrix is organized into connective tissue, which is made of collagen and elastic fibers. It provides physical protection and resistance to compression and stretching. The basement membrane determines basically the cell shape. It provides a storage for growth factors and chemotactic peptides. It also acts as a surface for migrating epidermal cells after the injury. This is an example. This is right here. This is a heart, and here we see fibrin deposits in the pericardial sacs right here. So fibrin deposits. We have these fibrin organizations may lead to collagen fibers, and here we see the heart with collagen calcification. So it's like an example of the organization of collagen. This is another example. We have a traumatic neuroma right here, and we see the original nerve entering to the neuroma. The nerve is surrounded by the blue collagenous tissue right here in the The reorganization and remodeling of the extracellular matrix have some key players. We're going to see neutrophils, serine proteases, and matrix metalloproteinases. These last ones are enzymes and are key players. They enable cells to migrate through the connective tissue framework by degrading the matrix proteins at the injury site. They play a big role in cell-to-cell adhesion, cell communication, cell growth, and during apoptosis, during remodeling. There is this molecule, the alpha-2 microglobulin, which is well known to inhibit this matrix metalloproteinases. Right here, this picture, this side shows us a healthy tissue, healthy enamel, healthy dentin, and a healthy gingiva, whereas in the other side we see some lesions and gingivitis and caries, carcinogenic lesions, and we see the presence of the matrix and metalloproteinases. There's other examples like in atherosclerosis right here, in liver pathology, emphysema, basically everywhere where there's injury, cartilage degradation, rheumatoid arthritis, and neuronal demyelination like in multiple sclerosis, right here in the blood-brain barrier degradation, and in blisters such as in bullous pemphigoid, right over there. Remodeling is basically when the injury site becomes a scar, and it's like it's when the equilibrium between the collagen deposition and degradation is restored. And so that's it for like the actions on the extracellular matrix. Now we're gonna see the last repair, the mechanism of repair, which is cell proliferation, and this basically takes place during the replacement of injured cells. And a big component is the formation of granulation tissue. This tissue is a specialized vascular tissue formed transiently during the repair process. Local and marrow stem cells accumulate in the wound and then differentiate into endothelial cells and fibroblasts. It is very important to point out that cells that are terminally differentiated, like neurons and myocardial cells, do not contribute to repair or regeneration. This is a foot ulcer covered by granulation tissue right here. Granulation tissue has two major components. We see basically angiogenesis and some cells playing key roles such as macrophages, which emerge from monocytes, and fibroblasts and myofibroblasts, which are descendants of the mesenchymal stem cells. It is important to know that at its peak, granulation tissue is the most richly vascularized tissue in the body. This is on a slide showing granulation tissue, and you can see right here in the arrows the thin wall vessels embedded in the loose connective tissue, and also the matrix full of mesenchymal cells and some inflammatory cells. Once repair has been achieved, most of the newly formed capillaries are reabsorbed, leaving a pale avascular scar. So here you see like very vascularized tissue and like red kind of scar, and this is like whitish pale with less vascularity seen. Okay, now we're gonna put all these mechanisms together. We're gonna add additional information, and we're gonna kind of divide the repair process into phases. So the initial phase of repair begins with hemorrhage into the tissues or any injury, and this is basically when a fibrin clot forms and fills the gap created by the wound. What happens basically is that the fibrin is cross-linked with the fibronectin by the transglutaminases right here in green. This cross-linking provides a provisional mechanical stabilization of the wound. Also during the initial stage, macrophages are recruited to the wound area, and they bind to fibronectin, facilitating phagocytosis. So you see here the macrophage secreting the proteolytic enzymes, and then actually eating the actual cross linkage of fibers.
Maria, how do those macrophages know to go to that site?
Because they're guided by cytokines. Yeah, it was at the beginning. I did mention it, I think, during cell migration.
Yes, I'm sorry.
No, it's fine. Now we're going to apply this initial phase into the skin. So what happens in the skin during this initial phase? So we're going to see thrombosis. We're going to see the formation of a growth factor-rich barrier having a significant strength. This is where all the platelets are going to come in and agglutinate. We're going to see some inflammation, which is going to be characterized by the macrophages, which will initiate the actual repair. And also we're going to see that during inflammation, necrotic debris and microorganisms are going to be removed by phagocytosis, by neutrophils doing phagocytizing. In the skin, we're going to see the re-epithelialization. This basically is when newly formed epithelium establishes a permanent barrier to microorganisms and fluids. This is an illustration of the initial phase in the skin. So we see all the key players. We see the neutrophil, the macrophages, all the growth factors, tumor growth factor, fibroblast growth factor, vascular endothelial growth factor, and everybody like going there. It's a nice representation. For the mid phase of repair, we have fibronectin, all the cell debris and the bacterial products being like the attractants for like all these cells. So here we can see the macrophages, fibroblasts being attracted to the actual wound site. As a new extracellular matrix is deposited at the wound site, the initial fibrin clot is lysed by a combination of extracellular proteolytic enzymes and phagocytosis by the macrophage. So here, once again, macrophage secreting these enzymes and phagocytizing the initial fibrin clot. We also see the deposition of a temporary matrix formed basically by protoglycans, glycoproteins, and collagen. The mid phase of repair in the skin, we see granulation. Granulation is going to be the specialized organ of repair and is the site of extracellular matrix and collagen secretion. The granulation tissue is going to be very vascular, very fluid-rich, insensitive, and resistant to infection. During the mid phase, we also see contraction, and this is when the fibroblasts transform to actin-containing myofibroblasts, and they link to each other and to collagen, and to type 3 collagen. Contraction is stimulated by tumor growth factor beta 1 or beta 2. And this is a visual representation of the mid phase. So you can see the blood vessels proliferating and the epidermis penetrating the epidermis penetrating the thrombus, but at the inferior side, and on top we're gonna see the scar. And the final phase of repair, we have the temporary matrix being removed by a combination of extracellular and intracellular enzymes, and the definitive matrix, which is going to be rich in type 1 collagen, is going to be deposited. And this final phase in the skin, it's going to be represented by the cross-linking of collagen, which makes it really strong, and remodeling. And this is when the wound site devascularizes and becomes distress lines in the skin. So we saw all these phases and key players in repair. Now we're going to put them together depending on the type of wounds that we're going to have. So we have the healing by primary intention. It's when you have a wound that is closely with opposed edges, minimal tissue loss, pretty small. Here we're gonna see little proliferation, cellular proliferation, and a minimal neovascularization, and the result is gonna be a very small scar, or maybe none. Healing to secondary intention is when we have a very big wound with edges that are apart and there is a substantial tissue loss. The key players here are going to be contraction, extensive cell proliferation, and extensive neovascularization, as well as the reepithelialization process. Also, granulation tissue it's going to be reabsorbed, but it's going to be replaced by a large scar, which is not, it's going to be functional, but it's not going to be very pretty to the eye, so it's going to be aesthetically unsatisfactory. So we saw that healing is the outcome of an injury. We saw the steps of repair, and now we see how repair would end up, either by regeneration or fibrosis. So regeneration is the restoration of original tissue matrix and architecture. Here we see like this finger, it has a big wound on it, and we see like the regeneration is complete, like it looks like nothing happened. And basically this process is done by perfect stem cell differentiation, and it's favored when the matrix composition and the architecture are unaltered. So what happens if the matrix is altered? The wounds are not going to heal, and this is going to be due to either excess proteinase activity or decreased matrix accumulation and altered assembly. Okay, we also are going to have fibrosis, or we may have fibrosis, which is scarring and collagen deposition. Here we have an example. This is a slide of myocardial infarction tissue, and the asterisk is going to show like mature fibrosis, and then underneath the arrow you're just going to see that some disrupted myocardial cells. This is an example of chronic fibrosis. And chronic fibrosis is when we have excess collagen formation. This slide represents chronic hepatic injury, and here in pink we see the regenerating nodules separated by fibrous bands, and in blue we see the bands of connective tissue. For scars, we have the keloid scars. These are thick scars. They may occur after burns or radiation. They may expand beyond the wound margin, which is like a very important thing to note, and they may be itchy or painful. They also may affect the skin movements. This is an example of a keloid as a reaction to having the earlobe pierced, and these keloid scars are more prone in dark skin or black individuals, and they have a tendency to be hereditary. Right here we have another example of a keloid scar 6 months after surgery. And we also have hypertrophic scars, which are elevated scars. They're typically red. But the difference between these ones and the keloids is that these ones are confined. So these do not expand beyond the wound margin. They're not painful. Here we see on a slide, this shows the epidermis right here, and you see the really thick dermis, and right here the collagen bundles with random orientation and tons of cells. And just to conclude, we see the overview of what we covered today once again. So we had an injury, then there is repair, inflammation kicks in. We can either have regeneration or fibrosis, which may lead to scars. That's it.
Thank you, Maria. We have time for a couple of questions. Any faculty members have anything they would like to ask?
That you don't feel like there is no
No nerve termination.
No nerve termination. so you don't feel. It's insensitive. So
It's been perfectly clear. Thank you, Maria.
thank you.
No, it's fine. I'm just gonna let it run.
I guess I might as well start reading the title to the next talk because it's quite lengthy. Giulio Scardini is going to present, if I might have your attention, please. He's going to present a paper, the title of which is The Lack of Association Between Common Polymorphisms of Epidermal Growth Factor Receptors and Non-Syndromic Cleft Lip with or without cleft palate. Quite a mouthful.
Yes.
Giulio,
Thank you. All right, today I'm going to discuss what Dr. Hada just mentioned, the lack of association between common single nucleotide polymorphisms of epidermal growth factor receptors and non-syndromic cleft lip with or without cleft palate. This study was published in the International Journal of Pediatric Otorhinolaryngology by Italian scientists from the University of Bologna. What is cleft lip and palate? Cleft lip are birth defects that affect the upper lip and the roof of the mouth. There are three variations under the cleft lip. One is the unilateral incomplete, which is basically just a simple cleft on one side of the lip. The unilateral complete, which is a little more severe, it goes all the way up into the nose. And the bilateral complete, which is the most severe of all the three, which affects both sides of the lip and goes all the way into the nose. Cleft palate also shows three variations. One is the incomplete cleft palate, which is the clefting of the soft palate and partially of the hard palate. Unilateral complete lip and palate, you have complete cleft of the palate, soft and hard, and the lip into the nose. And then you have the bilateral complete lip and palate, which is the most severe of the three, which is affects both sides of the lip and it goes all the way into the nose. Cleft lip and palate repair. Surgery to close cleft lip is often done when the child is between 6 weeks and 9 months old. So you shouldn't see children born in the United States who is over a year old with a cleft lip or a cleft palate. To repair the cleft lip, it only requires a simple surgery. Surgeons just stitch the lips together and that's basically it. Now, cleft palate is usually closed within the first year of life so that the child's speech development is normal. Sometimes a prosthetic device is temporarily used to close the palate so that the baby can feed and grow until surgery can be done. And this is what they use. This is a baby plate that it's used when babies have cleft palates. And this allows the baby to feed normally, because it kind of seals that the gap between the oral and the nasal cavity. Here I have before and after pictures of two children. This 3-month-old boy looks like he has a unilateral incomplete, right, cleft lip, and the little girl has a unilateral complete that looks like also that she has also cleft palate. This is 1 month after the surgery. This is how they look like, and later on, this is how they look like. Really, the scar really fades with time, and you can see the 5-year-old girl doesn't look like she had anything done to her lip. Now, CLP may not be life-threatening, but many functions can be disturbed because of the structures involved. These functions include feeding, digestion, speech, middle ear ventilation, hearing, respiration, facial and dental development. However, feeding can be life-threatening because children with cleft palate, if they don't have baby plates, they have the tendency to breathe in milk, and that can get into their lungs, and that could be life-threatening for them. These problems can cause emotional, psychosocial, educational difficulties. This is particularly important in underdeveloped countries where children don't have access to healthcare. In certain cultures, they believe that those children are cursed, so they're shunned by society and by their parents, and that's very significant, a problem for them. All right. The etiology of CLP is theorized to be a combination of factors associated with genes and environment. Genetic predisposition to environmental factors include anti-epileptic medications taken in pregnancy, smoking, alcohol, infections, folic acid deficiency, and vitamin A toxicity. Just 1 minute. Cleft lip and palate is divided into syndromic or classified as either syndromic or non-syndromic. Syndromic is when children who already have a condition, a pre-existing condition, such as these ones here, like Down syndrome, fetal alcohol syndrome, Stickler syndrome, Treacher-Collins, and all the others, are predisposed to having cleft lip. The non-syndromic is not connected to an existing condition, but it is associated with single nucleotide polymorphisms within specific genes. And we're going to be looking at the gene that's coding for that, for the receptor in this study. What I'm showing here to you is the transforming growth factor A. This is a transmembrane protein expressed at the medial edge of the epithelium, and its receptor is the epidermal growth factor receptor, the gene under study, or the protein under study. This is a ligand, a cytokine that's going to bind to the receptor, and this will lead to a cascade of reactions which ultimately results in the normal development of a lip and a palate. So this is what happens. Transforming growth factor binds to the epidermal growth factor receptor. This leads to a cascade of reactions that will result in cell survival, proliferation, and ultimately in the normal development of the lip and the palate. Okay, these are the stages of palatal development in mice, which are pretty similar to what humans go through in fetal development. You have a vertical growth, elevation, adhesion, and fusion. Disturbances in any stage during palate development, including defective palate shelf growth, failed or delayed elevation, and blocked fusion leads to cleft palate with or without cleft lip. So any disturbance on this process here will lead to the cleft lip and palate. All right. What is single nucleotide polymorphisms, or single nucleotide polymorphism? Is a DNA sequence variation occurring when a single nucleotide in the genome differs between members of the species or between paired chromosomes of an individual. It's basically a change. This is a gene, and what's happening, a single nucleotide polymorphism is basically a single change from one nucleotide, from this combination to this combination. This change could affect the amino acid sequence of a protein and change the function of that protein. And this is what they're going to be looking at in the epidermal growth factor receptor, the gene that codes for that receptor. They're going to be looking at this the polymorphism within that gene. Okay, the epidermal growth factor receptor is a transmembrane glycoprotein which mediates the effects of the transforming growth factor alpha. The transforming growth factor alpha, as I mentioned, is the ligand that binds to this receptor that leads to the normal development of the cleft, of the lip and the palate. There are 4 members within this receptor family: the EGFR, ERBB2, ERBB3, and ERBB4. Okay, now let's go on to the study, but just to review what I just said. Scientists here are investigating the association between the polymorphism, the single nucleotide polymorphism within the gene that's coding for this receptor, the epidermal growth factor, and they're looking at if there is any association between this and non-syndromic cleft lip and palate. Materials and methods. They recruited 239 Italian children with the sole clinical feature of CLP and their parents. Of the 239, 91 cases were dropped because they show a positive history, a positive family history of cleft lip and palate. And they were, and what this means is that other relatives share the same malformation. So they discarded this group here, these 91 cases, because they could explain this by normal Mendelian inheritance pattern. So they're not interested in this. They're interested in the other cases, the non-syndromic, non-familial cases. And the remaining 148 cases were the ones that they actually studied. And they were classified as non-syndromic because they did not show positive family history. All right, the markers are the single nucleotide polymorphisms in the genes that are going to code for this receptor. And they look at 3 of the members of the family of receptors. And the single nucleotide polymorphism, the GA located on the EGFR exon 13, the AG which maps onto ERBB2, and AT which is located in ERBB3. No polymorphism was available for the 4th component of the EGF receptor family, the ERBB4, and therefore this gene was not investigated. Restriction fragments were separated by electrophoresis on 10% polyacrylamide gels and visualized using staining. Okay, the statistical analysis. They use the transmission disequilibrium test, which is a very complex test. And what I'm going to tell you is just a simplified definition of what it is. It was used to prove the linkage hypothesis in the presence of an allelic association. TDT is a family-based association. This is an association study. Test for the presence of genetic linkage between a genetic marker. The genetic marker is the single nucleotide polymorphism in the gene that codes for that receptor, and a trait, the cleft lip and the palate. It detects genetic linkage only in the presence of genetic association. TDT considers parents who are heterozygous for an allele associated with disease and evaluates the frequency with which that allele or its alternate is transmitted to affected offspring. Here it is. I only want you to really focus on one column here, and I want you to focus on this column. Basically, what they have here, they have the 3 members of the receptor family and the genes, the single nucleotide polymorphisms that they were investigating. And I want you to just focus on this. This is the transmission versus non-transmission of this polymorphism to offspring. As you can see, if you can look at the numbers, you can see that they are basically equal. There is no clear indication that the genetic polymorphism that it's causing cleft lip and palate in the children really it's really coming from their parents. So there's no clear evidence just by looking at this number, the transmission, because these are very, there's no real difference between the transmission and the non-transmission. All right. So this is a summary of what they are concluding. They're concluding that a ligand like transforming growth factor alpha binds to an EGFR, or the epidermal growth factor receptor, and one of them, or all of them, may have single nucleotide polymorphisms. They're saying that that interaction leads to normal cell growth, proliferation, differentiation, which leads to the normal development of the palate and the lip. But what they're saying is that this does not lead to cranial malformation or craniofacial malformation, which does not result in a cleft lip or a cleft palate. So what they're saying is that, and they stated that no single marker resulted as being significantly associated with CLP. Indeed, no significant departure from Mendelian inheritance pattern was observed. Results suggested that the investigated polymorphisms did not constitute a risk factor for cleft lip and palate. The results indicated that no single nucleotide polymorphism, nor any combination of them, act to influence the disease. However, given the clinical and genetic heterogeneity of the disease, further investigations involving different populations are needed to verify whether the epidermal growth factor receptors play a role in cleft lip and palate onset. So what they're basically saying is that even though they didn't find an association between this single nucleotide polymorphism in the gene that codes for this receptor with cleft palate or cleft lip and palate, they're not saying that it doesn't exist. So more study is kind of required here for them to be able to really say that that association really exists. And that's the end.
Questions? Dr. Mariasi?
Yes.
I've been looking at some of these growth factors in the lung, and there's a body of evidence that implies that many of these growth factors are stored in the basement membrane of the particular scaffolding that you're building. And any additional misdeeds in the basement membrane tend to interfere with their action, because if they're not stored, then they cannot act. I was just wondering if these people discussed the idea what other factors might be involved here. If this by itself does not show any relationship, maybe other items that are in the process of delivering these signals might be involved.
They discussed the transforming growth factor beta 3 as being involved in this, and beta 1, beta 2, and beta 3 being the most important of all of them. And transforming growth factor also, another transforming growth factor, but I don't recall which one it was. But there are others that they mentioned that it's associated with normal development that may be also, if disturbed, could lead to cleft lip and palate.
I meant if any storage ideas of these factors as they're being ready for action is involved in this in order to explain why there's no association.
They didn't mention any storage, no.
Thank you.
You're welcome.
What were your expectations about the influence of seemingly minor mutational changes occurring, single nucleotide polymorphisms? Would you expect the results they got?
Yes, because there is something that they mentioned that I didn't really say, that these receptors dimerize into dimers, right? They're single receptors that dimerize. And because they do that, so this polymorphism that's happening, that's changing maybe the amino acid sequence of the protein, is not really changing their function because of the fact that you have another one coming in. That's basically why I kind of understood why that wasn't a lack of association.
Right.
That makes sense.
Makes sense to me.
Have any of these the cleft palate issues, have they ever been tied to any other developmental issues, neurological?
He just said this. Syndromic means he presented very well that one. You didn't pay attention. All those syndromes are, like he mentioned at least 3 of them, they may have cleft lip.
Okay. I apologize, I didn't see it.
It is for everyone.
A lot of these are neural crest derived problems. The neural crest comes in that area, fills the mesenchyme in
Right.
that area, and bones and everything. And so a lot of them are linked with like heart defects. As the neural crest comes in and gives off some of the cells there for the valves, and then it continues upward. There's a migratory path into this area, so that you have a lot. The outside is from, this is a big sort of pillows that come together and fuse right here, right along those little ridges you have. That's why it's very common. The lateral cleft palate, cleft lip is very, very common because it comes in here and then squeezes right here. This little piece here is separate. So here, until you get 2 little pillows coming in and they'll fuse here. If there's not enough mesenchyme, which is no crest, they won't fuse. Underneath, you've got shelves coming in, bone shelves closing up together with a little corner here. The same part that's here makes a little corner with the eye teeth. And they all come in. That's why a lot of the cleft, or there are a number of people born without those teeth, because that's right at a fusion line.
The importance of these studies are coming from the fact that cytokines were never thought to be involved in this type of embryonal development. And for many years, cytokines were considered just the mediators of the inflammation. And during the last 10 years, they saw that every place cytokines are the main acting factor in normal development, and they are in inflammation, in neoplasia, in atherosclerosis, bronchial asthma. So cytokines are, in this very moment, the most important acting factors in the human body, and obviously in animals, as you saw, many studies are done on mice. But another issue he mentioned very nice, the precocity of the need of surgery. In my time, there was they said, no, you cannot touch a baby until he is minimum like 3 months old. So remember, he said 6 weeks. So the concept of when the surgery is the best to be performed changed dramatically. And this is very important because this shows how quick the technology helps science and science helps technology.
And basic science helps all.
Yes. Dr. Marius, he helped us to survive.
And you're not whistling Dixie either.
Any faculty have any comments, any any messages from Nice job.
Thank you.
Kind of shaded in the back.